Welcome to this tutorial. In this session, we're going to look at nucleic acids. We're going to understand what nucleic acids are, what make up nucleic acids. We're going to understand the two categories of nitrogenous bases and so many things about nucleic acids. So stay tuned as we discuss this topic in details. Let's begin by knowing what nucleic acids are. So we are looking at nucleic acids. For better understanding of this topic, we need to grasp the concept or the basics about nucleic acids. What are really nucleic acids? So nucleic acids are just compounds or components that make up the DNA and the RNA. Or we can also say they are the compounds which make up the genetic information. So we are saying compounds or molecules that make up DNA or RNA. Of course, we know that for DNA to be made into RNA, it has to be transcripted. The process of conversion of DNA into RNA, that is known as transcription. Even before it can be formed into proteins, because proteins are so much essential in forming the genetic information. So we need to understand that. Now let's look at what make up the nucleic acid. What are the components which make up the nucleic acid? So we need to understand that the nucleic acid are made up of the nucleotides. Let's understand what nucleotides are. So nucleotides are just the compounds which are composed of different molecules. I'm going to talk about the three important components of the nucleotides. So I'm going to say nucleotides are composed of. So we need to know the three important components of nucleotides. The first one is the phosphate group. The second one is the pentose sugar. Pentose. The pentose sugar. And we understand to say the pentose sugar is a furanose ring, right? And the third component is the nitrogenous base. Nitrogenous base. I can abbreviate this as NB, the nitrogenous base. So these are the components which make up the nucleotides. Now let's look at a situation whereby you've removed the phosphate group. We remove the phosphate group from the nucleotide. What are we going to form? So if at all we remove the phosphate group, then we are going to remain with the pentose sugar We'll just remain with the pentose sugar and the nitrogenous base. So if we just have the pentose sugar and the nitrogenous base, we've removed the phosphate. That will be called a nucleoside. Nucleoside. It will no longer be called a nucleotide because it has lost the phosphate group. So what are we saying? So what we are saying basically is a nucleotide can form an equation minus a phosphate group is simply equal to a nucleoside. So this is an equation we formed. A nucleotide minus one phosphate or minus the phosphate group gives a nucleoside. Now let's understand the components now in detail. We'll start with the pentose sugar. We've said pentose sugar is one of the major components of the nucleotide. So how is this pentose sugar? So, of course, it's a furanose ring. So it will have a 5-member structure. That is a furanose ring. Like that. So there will have OH. We've done the carbohydrates. Like that. So, of course, we need to understand how to draw these ring structures. So there we'll have CH2OH, H. So this compound, which I've drawn here, this is a ribose. A ribose is a 5-member ring structure. It's a furanose. Okay? Furanose meaning a 5-member ring structure. It is called a ribose because all the hydrogens are just maintained in this ring structure. But let's say a situation whereby we tend to lose that hydrogen. Let me move. We tend to lose the oxygen rather. So all the oxygens are maintained. Instead of having oxygen there, we have the hydrogen. So because this compound has lost one oxygen, it becomes deoxy. So deox means there's a loss of oxygen. So it ceases to become ribose sugar. Instead, it will be called now as deoxyribose. So this is very important for us to appreciate. Now let's look at the complete structure of a nucleotide. So it will be formed by this pentose sugar. The pentose sugar will be a backbone of it. Then there will be some condensation reactions which are going to occur at that carbon point, and also other condensation reactions which are going to occur at that point. Now let's see what is going to be formed, the structure now of a nucleotide. Let's look at nucleotide structure. So when you're looking at the structure of a nucleotide, let's have the pentose sugar, like that. Okay, so this is the pentose sugar we have. at that point, there will be a certain condensation reaction. That is where we are going to attach, because there was OH there will be a condensation reaction. So that's where the reaction between this ribose is going to react with the nitrogenous base. So we're going to have a nitrogenous base there. Then, that is at carbon number 1. At carbon number 4 there will be another reaction which is going to take place. Okay? In fact, let me say just at carbon number 5, because that is where we had 2CH or H. Okay, so at this point, there will be a reaction between this betel sugar and the phosphate. So we'll have that. So this is where the phosphate is going to be. This is where the phosphate is going to attach itself. So this is the phosphate. Okay? Now let's look at the reactions. Of course, let me say this is a ribose sugar, not a deoxyribose, right? Because of OH there, it's a ribose sugar. Okay, so let's look at the reactions, or the bonds rather. So the bond which is going to exist between this ribose and also the nitrogenous base, that one we're going to call it as the N-glycosidic bond. We understand to say the glycosidic bonds are the bonds which are found in carbohydrates, right? Because the ribose sugar is part of the carbohydrates. Okay, it's a carbohydrate, so it's going to form a glycosidic bond with the nitrogenous base. Then between this ribose sugar and also the phosphate group, there is a bond which is going to be formed. So this bond which is going to be formed is the one which we are going to term as the phosphodiester bond. So here there will be a phosphodiester bond. Let me write it. So there will be a phosphodiester bond. So it is very important to understand that. But even before the phosphodiester bond is formed, phosphodiester bond, it means there's another phosphate here which has been bonded, okay, to this. Then there are two phosphates. There are two phosphate bonds. That is when it's going to form phosphodiester bond. But before we can make that, di means two. So let's just call it a phosphoester bond, or a phosphate ester bond. A phosphate ester bond, because it has not yet, there are no bonds which are yet formed. There's just one bond. That is a nucleotide. So let's now understand something interesting here. So I'm now going to look at the nucleic acid structure. So nucleic acid structure. Okay. Let's understand the nucleic acid structure. Okay, this is what we want to understand. Okay, so let me just wrap. So this is what we are going to have let's say we have a furanose ring there, one furanose ring. Then there's a bond with the phosphate there. Then it forms that attaches to another nucleotide. So again, there's a bond here with the phosphate. Then it attaches to another nucleotide like that. So this is what we have. Then let's say these are the nitrogenous bases. Even there we have the nitrogenous base. Nitrogenous base. Even there we have the nitrogenous base. Then of course let's say another phosphate group there. Okay, so let's understand something interesting here. Of course, we understand to say the bond between this pentose sugar, which is the ribose, and the nitrogenous base, we've called that to be the N-glycosidic bond. So we understand this bond. It is N-glycosidic bond. N from the nitrogenous glycosidic bond. Then the bond between, okay, so we're going to have this bond, okay? So one end is the 3'. This 3' is connected to the 5' end. So there's a 3' there because at carbon number 3, that's where this bond is being formed, and that's carbon number 5. So there is a phosphate ester bond there, even the phosphate ester bond there. Because there are two phosphate ester bonds, we are going to say the bond which is existing here is the phosphodiester. Phosphodiester bond. The diester, di is coming about because there are two ester bonds. There are two ester bonds. The one which is linking the 3' end to the phosphate group, and another one which is linking the 5' end to the nucleic acid. So it is very important for us to understand that. The point I'm trying to put across is that there is this one bond which is at 3' end linked to the phosphate group, and another one which is at 5' end linking to the phosphate group. So very important for us to understand this. Now let's look at the two categories of nitrogenous bases. Categories. Categories of nitrogenous the categories of nitrogenous bases. Let's look at the categories of nitrogenous bases. So there are only two categories of nitrogenous bases. The first category, it is the purines. So what are purines? Purines are the double ring structures or two ring structures. They are two ringed structures. Those are what we call purines. So how are the purines? They are like this. We can have a 6-membered ring like that, joined to a 5-membered ring. Okay? Join to a 5-membered ring, let's say like that. So one is a 6-membered ring, the other one is a 5-membered ring. They are double ring structure. Those are what we call purines. There are 2 purines. So we are looking at purines. So we need to understand that there are only 2 purines. There are 2 purines. One is known as adenine. The other one is known as guanine. So adenine and guanine are the 2 purines. So let's look at where these are found. Let's look at where they are found. The adenine, we need to understand that adenine is found in both RNA and DNA. In DNA you'll find adenine, and in RNA you'll still find this adenine. When you're talking of the guanine, it is also found in both RNA and in DNA. So it is very important for you to understand this. So how can you remember this? You are saying purines, it is the examples. How can you remember the examples? It is adenine and guanine, right? So you can make a mnemonic memory head whereby you say this is maybe pure as God. Pure as God. Or maybe you can say Pentecostal Assemblies of God. Okay? Just to remember, as a memory aid, just to remember. First, just for you to remember that we are talking about purines. They are formed of, or they are made up of adenine and guanine. So that is very important. Let's look at another category of nitrogenous bases, which we call pyrimidines, or pyrimidines if you want. Pyrimidines. So what are pyrimidines? Pyrimidines are just single, one-ringed structure. Okay? They are just single-ringed structure. Single-ringed structure. So this is one important thing we need to understand. And other than that, they're made up of the 6-ring structure. It is a 6-ringed structure. So if at all you were asked to draw the 6-ring structure, should be something of an hexagon like that. Okay? So that would be the 6-ring structure, also known as the pyrimidine. Now let's look at examples. So for pyrimidines, there are 3 examples. One, it is the uracil. 2, it is the thymine. Thymine. If you say thymine, thymine is a vitamin. Okay? It is something else. So you can pronounce it as thymine, or you can say thymine if you want, as long as you meet in spellings. Then you can talk about the last one, which is the cytosine. One thing you need to understand is that uracil is only found in RNA. Thymine is only found in DNA, but the cytosine can be found in both. Okay? This can be found in RNA and in DNA. Now, to remember the pyrimidines, you can also use maybe a memory head also to remember this. You can say maybe put C, where P is the pyrimidines, U is the uracil, T is the thymine, and C is the cytosine. So it is very important for us to understand this even as we talk about the nucleic acid, because this information is going to be very much essential. Okay, so let's look at what is known as the Chagall's rule. What is it about this Chagall's rule, or what do we need to understand about this Chagall's rule? Okay, so about the Chagall's rule, we need to understand that, or we need to grasp the concept where it states that when DNA, or the DNA from any or from all organisms, right, it should have a 1-to-1 ratio in terms of base pair rule. Okay, so that is the Chagall's rule, okay, which we need to understand. Okay, excuse me for that. So what we are saying is that according to Chagall's law, DNA from all organisms not from one organism or from two organisms, but from all organisms, it should have this important ratio, 1-to-1 base pair rule. That is what the Chagall's rule states. So the DNA from all organisms should have only 1-to-1 base pair rule. If you understand that, that will be very important. And it will be very essential for you to understand that. So the next thing which we are going to talk about, let's look at some few structures. Let's say you have something like this. Then there you have the OH. Then there we have of course, the nitrogenous base. Let's say that is the adenine. Then of course we have the phosphate. Now, because in this compound we only have one phosphate group, so the name will start with the nitrogenous base, of course. So in naming, you need to start with the nitrogenous base. So the adenine will change to adenosine. We are saying adenosine because at this carbon there is OH. This is a ribose, okay? So it will be adenosine. Because there is just one phosphate, it will be adenosine monophosphate. So we are calling mono means one. Let's have a situation whereby, what about this same structure? We've added now 3 phosphates. Even the name changes. The name will change because there are 3 phosphates now. We are going to say this will be adenosine. Because of 3 phosphates, it will be triphosphate. Tri is coming from having 3 phosphate group there. So this is what is breaking up the triphosphate. So very important for us to understand. Okay? So it's very important for us to appreciate this concept. And you see, this one, which we are calling adenosine triphosphate, this is the one we call ATP. The common form in which energy is stored, right? Energy is stored mostly in ATP, form of ATP. So this is what we call adenosine triphosphate. It's made up of 3 phosphate, the adenine group, and also a ribose sugar. Okay, now let's look at another structure. Let's say instead of having the adenine group, you decide to have a different nitrogenous base. Let's say you have a different nitrogenous base. Okay, you have this. Instead of having adenine there, let's say you have guanine. Then of course there is a phosphate, a phosphate, a phosphate. What will be the name? So even the name will change. So that's what we are calling it adenosine because of the adenine. So this will be guanosine. To be guanosine because of guanine. Because there are 3 phosphates, guanosine triphosphate. So this is the one we call GTP, guanosine triphosphate. It is very important for us to appreciate this concept. So one thing I need you to take home here is that if the sugar which is being used in this structure, if the sugar which is being used in this structure, let's say it's a ribose sugar. So the compound or the nucleic acid which is formed is the ribonucleic acid because of the ribose sugar. Okay? It's very important for you to take home this. So let me just a minute, I write something. Alright, let's continue, please. Sorry for that. Okay, so we are saying if the sugar which is being used, let me write it here. So if the sugar which is being used is a ribose, the nucleic acid which we are going to form is the ribonucleic acid, which is known as the RNA, ribonucleic acid, because of ribose there. That is where ribo is coming from. But if the sugar which is being used, the pentyl sugar is deoxyribose. Let's say it is deoxyribose, right? It has lost 1 oxygen. So this one, the sugar which it is going to form is deoxyribonucleic acid. So this is what is going to be formed.